All right, hi everybody. So I'm sorry about this, but for reasons that my brain only knows of itself, I forgot to record this morning's lecture on linkage. And so I know many of you are unable to attend it in person, so I think it's important that I do this again, uh, so that you get what you need from this particular lecture. Oh, damn it. And also, linkage is it's a tricky topic, right? And so I think it's not going to be the full lecture because dinner's in like an hour or so, and I'd like to eat it. But I hope to cover all of the kind of salient points, so at least you know what questions to ask next class. And also, I have a series of um, pictures that were taken from today using examples and working through particular linkage problems. And I also have the chat session from today. So that'll also, I mean, there's obviously a lot of chit chat and admiration of my cats, but there's also a lot of work on the calculations. Oh, that's kind of weird. Yeah, anyway. Okay, so linkage, right? We're working from PowerPoint 9. And so really what linkage is about is distance. Distance and random chance of recombination. So, oh, before I even get to that point, linkage is about genes that are on the same chromosome and maybe affecting one another when it comes to inheritance of alleles. So, the law of independent assortment, one of Mendel's three laws, states that what allele you get in a certain gamete, again by chance, remember the law of segregation, has no effect on which allele you get for another gene. So the two genes essentially act independently, or loci, if you wish, right? So there's no interaction between the two. So they're essentially random chance applies to this one and this one, separate from each other. Okay, so linkage breaks that. Essentially, linkage is saying that what allele you get for this gene does, to some degree, and that degree can vary depending on distance, affect what allele you get for this gene. So if you have a recessive allele locus A, and that locus is linked to locus B, then whatever allele is present on locus B, wherever you find the recessive allele A, you will tend to find those two together. Right. So another kind of more general term for linkage is that alleles are traveling together. If you find one particular allele, you're more likely to find another, right? It's not random chance. Okay, so the big deal in linkage is distance and probability, or just random probability. So I use an analogy today. I'm not sure it was a very good one, but it kind of worked, which is it's the probability of recombination occurring is the same for any particular stretch, with some exceptions, and you can see those at the end of the PowerPoint. So the bigger the stretch, the greater the probability that recombination occur. So my analogy, again, not a very good one, was that it's like running across a road blindfold in traffic. If you run across a single lane road blindfold in traffic, you have a certain probability of being hit by a car. If you run across a two-lane road with the same amount of traffic, your probability doubles. 3 lanes triples, right? And so essentially those two, by and large, and again, there are some exceptions, but it mostly holds true. The bigger the distance between two genes, the higher the chance of recombination until they get far enough away that recombination occurs all the time. These genes essentially act independently or sort independently. So they are not linked. So I'm just going to, during this, because I'm not much of an artist and I've already done it once today, I'm going to refer you to a series of pictures. So these are already uploaded onto Blackboard. These are made with my little miniature whiteboard. Hang on, I can actually share that with you, can't I? Let's see. Okay, so essentially that's what this picture is showing. If you have two genes or loci, again, and it doesn't matter, right? They're just places on the chromosome that are distinguishable from one another, essentially. If those two loci are far enough apart that there's always recombination occurring between them, and that's what that X shows you, right? Is that where you have your homologous chromosome together, this one in the middle, these two in the middle, the non-sister chromatids, are recombining or crossing over every meiosis between those 2 points. Now that doesn't mean their recombination is occurring between any 2 point on the chromosome, right? Just for the two that we're looking at. And so essentially, if there's always recombination between the two, you end up with 4 different possible gametes or 4 different possible chromosomes, which is essentially the same thing, right? Because each gamete only gets 1. So you have at the top, this one here, hopefully you can see what I'm circling, a parental chromosome. That's one of the chromatids that's on the outside of that pair, right? So say this one here. That will have the same combination of alleles as one of the parent chromosomes. You'll also find another parental chromosome, this one down here. Obviously, this is in an individual that's heterozygous, both loci. Forgot to mention that. You'll also have another parental chromosome which has the recessive A and recessive C allele. You'll see that down here. That's the other parental chromosome also on the outside of that homologous chromosome pair, right? So really it goes back to the mechanics of crossing over during prophase 1. However, again, if these genes are not linked and recombination always occurs, so essentially that it's just distance, then you'll also, every meiosis, get 2 new chromosomes. And these are going to be mirror images, right? And so essentially, if you follow kind of my cursor, we start here on the top chromosome, we go along, and now we cross over onto the other chromosome, and we have the recessive C allele, right? So this is a different combination to what was seen in the parents or the parental chromosomes. And because recombination generates a mirror image, you do the same with the other one. So you start down here on the other homologous chromosome that has the recessive A allele and zip up and again onto the paired chromosome and you have the dominant C. That will give you the one down here. So if there is no linkage, you'll always get those 4 possible chromosomes and therefore the 4 possible gametes, which will then, through sexual reproduction, produce the offspring. And so that gives us a 1:1:1:1 ratio of possible gametes. Keep that in mind, it's really important. Okay, so this is if there's no linkage. That's one extreme. So that's when these genes or loci are so far apart that recombination always occurs between them. Okay, what about if they're so close that recombination never occurs, right? They're literally right next to each other or even like on top of each other. Now we have what's called completely linked. So I drew this over the other diagram so you can still see genes A and C. You can still see the ratio for genes A and C. Now we have no recombination between these two genes. They're so close that for the, let's say, the cross that we see or look at, you never see a recombination event between these two genes in any of the meioses that generate gametes for that particular cross. Now, obviously, that's slightly context dependent, because if you look at 5 million offspring, you may pick up a rare recombination event. Whereas if you only look at 1,000, you won't, right? So again, it's just mathematical probability. So now we have them so close together, there's never recombination event. And so there's no X here, right? So in the offspring, you'll get this chromosome for AB. Remember, we're just looking at AB here, right? Or you'll get this combination of alleles. So even though recombination may be occurring elsewhere on the chromosome, which essentially, generates these other combinations between A and C, between A and B, there is never recombination. So all you ever get are dominant A, dominant B, or recessive A, recessive B, right? So these alleles or these genes are linked, or rather, these alleles travel together because the genes are linked. Again, it's very much context specific. So A and C are not, A and B are, right? So you have to be very careful. What loci are you referring to when you talk about linkage? And so in terms of gametes, now you have a 1 to 0 to 0 to 1 ratio. So 1 because they're either all dominant, 0, 0 because you never see any recombinant chromosomes, 1 because the rest are recessive. Okay. Let's see what I've got next. Okay, so essentially we can use that chance of recombination to work out distance. And so if we go back here, we can see that, again, they're so far apart, there's always recombination. But as you get closer and closer and closer, at some point you hit a distance where in one or occasional meioses, recombination does not occur. So now you see a phenomenon when this is a hallmark of linkage, where there are more parental chromosomes or parental gametes than recombinant. And so if you see this, that's a sign that linkage is going on, that these two genes or loci are close enough that recombination doesn't always occur between them. Because again, chance of recombination is proportional to distance. And at some point, that you get down to chance of less than 1 or less than 100%, right? And so now you'll start seeing an excess of parentals, and that's key. Really important to remember. Obviously, if you go to the full extreme, right, this is an absolute excess of parentals. There are no recombinants whatsoever, but you'll rarely actually see that experimentally, right? Genes have to be very, very close together for that to happen. So instead, what you're likely to see is something between the two. And we can use that to calculate distance, right? That proportionality between recombination and distance between two loci. So essentially what we spent the rest of the lecture doing was going through a bunch of examples. And so what we're going to do first is we're going to go through, a 2-point cross, which is comparing, using recombination between two loci to calculate distance. And we'll also then go on to 3-point crosses. So another thing that I covered briefly during the lecture was the concept of linkage phase. And so one thing that I stress is that you cannot assume anything when you look at recombination and a 2 or 3-point cross. You have to work by the numbers and what information is given to you. So Linkage phase is important in 2 and 3 point crosses because it basically determines what the recombinant chromosomes are going to look like, right? Oh yeah, I did do that right. Just looking at something there. Okay, so this individual is heterozygous. This individual is in the trans linkage phase because it has essentially one of both, or alleles that are different in terms of their dominance for each loci. So this is a recessive allele, this is a wild type or dominant allele. And the mirror image on the other chromosome, which is written down here, is dominant for Z and recessive for X. So it's important to know that. Let's see if I've got some other example. Okay, so here's another example. This is where the linkage phase is in cis, C-I-S, and that means same essentially. So, or coupling or whatever you want to call it. So you have both recessive alleles on one chromosome and both recessive dominant alleles on the other chromosome. In terms of genotype, this doesn't make a hoot of difference, right? You still have the same genotype. But in terms of linkage, it makes a big difference because now you can see that the wild type or parental chromosomes in this case, when there's translinkage, is recessive dominant and dominant recessive. When the genes are in cis linkage, now it's recessive and dominant are the parentals. So you can't assume that. You have to work it from the numbers. You may be given it, which would be nice, but there's no guarantee that you will. So you need to be able to work that out from scratch. Okay, so the first thing to do is figure out which ones are parentals, which ones are recombinant. And so if all the numbers are more or less the same, essentially you don't have linkage going on, right? Or it would be very, very hard to detect. It'll be very rare. If linkage is occurring, what's going to be in excess is going to be the parentals, right? So the flip of that is that the recombinants are the minority in terms of the offspring. So what we first do is look, okay, what are the smallest numbers here? And this holds true for 3-point crosses too. The smallest numbers are these ones. Now we know because we've been given the linkage phase of the parent, we know that these are recombinants anyway because they're different from the parents. But if we weren't given that, we would know that these are recombinant chromosomes because they occur at the lowest frequency. Okay, so even if I, kind of put my hand up there and block this off while I was describing it, you'd still be able to figure out what linkage phase the parent was in purely based on this, right? So this is a recombinant chromosome, has 2 recessive alleles. So if it's a recombinant, then the parent must be in trans, right? Okay, so what we do next is we add up all of the offspring, right? So these are the product of all of the meioses that we're looking at. And we want to find out how often recombination occurs in meiosis, right, to generate that distance value. So we add all those up together. That gets us 788 offspring. And now we add the recombinants together. The reason why we add them together is because recombination is a mirror image event. If you generate one particular recombinant chromosome, you have to have the other one present too. And you can actually work backwards from that, and I'll show you in a second. So anyway, those two together, that's 137. So the recombination frequency in total is 17 point that over the total, which is 17.3%. Do I have that on the other page? I don't. That's okay. So 1% recombination frequency equals 1 map unit, which equals 1 centimorgan, which is written lowercase c, uppercase m, right? Named after Thomas Hunt Morgan. So these two loci are 17.3 centimorgans apart. Now, if we wanted to work backwards, let's say I gave you 17.3% recombination frequency, and I could say, well, how many of each type of recombinant offspring would you expect to see? So given that the recombination is a mirror image event, well, I would expect to see for each mirror, essentially, half of the total recombination frequency, which in this case would be, what's that, 8.65%, right? So each different recombinant offspring, I'd expect to see about 8.5, thereabouts, percent of the total be that one, so the double recessive, or the double positive. I just realized I didn't write a plus there. Anyway, doesn't matter. Can't change it now. And then the percent each parentals would be simply 100 minus 17.3, which is what, 82.7 divided by 1. So approximately 42%, give or take 41, 42% of the offspring will be each type of parental. All right, hopefully that makes sense. If it doesn't, write it down, ask me a question next class. I want to make sure that you understand this. Okay, so next example, essentially we do the same, but now if you look, we've got X, now we've got YZ. So we've thrown another gene into the mix here, right? So this is our second 2-point cross. And we do exactly the same thing. This one is in cis, right? So automatically we know that dominant should be one of the parentals, and there it is. Dominant, 362 individuals, which is vastly in excess of any of the others. And we know that the flip side must be true, so that recessive Y, recessive Z is also a parental, also has a high number. So again, this holds true. Parentals are in vast excess of the total number of offspring. And so we do the same again. We add all these 4 categories together. That's our total, that's 802. And there are tons of these online, so really they're worth practicing, and I strongly recommend that you do, particularly for the 3-point cross. Now we add these two together, which is what, 59? Yeah, there we go, 59, and express that as a percentage. So you can see that there are fewer recombinants, right? And the total is roughly the same. So we know that these are closer together, and that's borne out by the recombination frequency of 7.4%. And you typically express it to 1 decimal place, right? So I'll make sure I remind you of that, when I ask you this in an exam, because you will get asked this. But just try and remember, it's always 1 decimal place. Just because even a, tenths of a percent or tenths of a centimorgan is still a reasonable size in genetic terms. Okay, so we can take those 2 bits of information and we can make a map, right? We know Y and Z is 7.4 centimorgans apart, and we know that Z and X are 17.3 centimorgans apart. We know that Y is not between Z and X because, actually, do we know that? No, we don't know that off of just this, but it's quite likely. We'd have to do another cross between Y and X to double check this map. And there's a good example of this map, of this kind of mathematical logic puzzle in the PowerPoint, where it asks you to sort out the order of A, B, C, and D. Right, so actually, do I have? Oh yeah, I got that up next. We can do that. So anyway, this is a putative map, right? This is a one of possible maps which, without any extra information, is perfectly valid. So then you can figure out, okay, how many recombinants am I going to get between X and Y would I expect to get if I looked at 1,000 offspring? right? And all you do is you just add those two numbers together. That's what's 27.7. So 27.7% should be recombinant or thereabouts, right? Which would be 274 offspring. Okay, so you can use these numbers to figure out gene order, right? And so there's a lot of crosses involved here. There's what, 6 in total between 4 different genes or loci. And so these numbers down here are percent recombination or recombination frequency. And what you can tell from the first three, anytime you see 50%, you'll never get higher than 50%, right? Because in any meiosis between two unlinked genes, you'll never get more than 50% recombinance, right? You just cannot, because the chromatids, remember, on the outside cannot recombine. So 50% is like the hard limit. Above that, you just don't see it because you can't. So anytime you see a figure of 50%, that means that those two genes are so far apart, you cannot see linkage occurring, right? They are unlinked, so you can't determine distance. We don't even actually know if gene A is on the same chromosome. It may not be. Right, we can assume it is, but we don't have any evidence for it being so. However, B, C, and D all appear to be fairly close together, right? And so you can use this. It's just a simple kind of math game, essentially, to try and figure out which must go next to which, right? So which is the closest? B and D, right? We've got B and D up here. So B and D is 10% recombination. And so then we want to figure out, well, where's C? So C could be over here to the left of D, in which case the distance between B and C would be greater than between B and D. And it is, right? It's 20. You can see up here. So if we have C over here, then C and B is going to be greater than D and B and C and D, right? But let's look down here and you find that C and D are actually 30% or 30 centimorgans apart. And so that tells us that B has to be in the middle. Handily, these two also add up to 30 as well, which is another clue. Right, so this gives us the gene order, D, B, C, or relative to each other, but it doesn't tell us which way round that order is relative to A. So it could be either D, B, or C C, B, or D. The internal distances stay the same. Which way round they are, we don't know which it is. And the only way of finding that out would be to do another cross with something closer, which is in between A and D or A and C, for example. Essentially to do another cross. Okay, so this is really laborious, right, to do all of these crosses. Because if you think about actually doing this in the lab, if you were working in plants or mice or whatever, you'd have to do 6 different crosses to get this information. And there's the risk of errors every time you do so, and they can kind of stack up. So it's much better to do fewer crosses, and the way of doing that is what's called a 3-point cross. And so this is an example of a 3-point cross. Now, I'll tell you in all fairness, you cannot rely on the gene order that you are given. This is not to trick you. It may be that the gene order is not known when you do this cross. You have to figure out the gene order for yourself. And I'll tell you how. So this is like a 3-step plan for 3-point linkage happiness, essentially, or 3-point linkage success in exams. So what you have to do first is you have to figure out which ones are the parents. And so this this drawing is taken from, where is it at? Down here somewhere. Here we go. This slide 22 in the PowerPoint. So the order that you're given these genes, les glossus in these, is not guaranteed to be correct. And you'll see that it isn't in a little bit. So, number one in your plan for success and happiness, figure out which ones are the parents. And again, this is like, I feel like some kind of dodgy internet telesales person saying, you just buy my thing for $29.99 and you'll be happy forever. Obviously it's only going to last for the period of this question in the exam or questions. So, where's that at? Yeah, dodgy internet salespeople. I'm not one of those, right? So anyway, step one of your plan for happiness and success, figure out which ones are the parents. And because this is linkage, you know that the parents are present in excess. That's not the band. Oh, geez, no, I don't want to go there. You'd have to be at least probably my age or thereabouts to get that joke. But anyway, it doesn't matter. Makes me laugh. The parents are going to be present in excess. So look for the highest numbers. Which one are those? Those are these ones down here, the triple recessive, and the ones up here, the triple dominant. So that tells you that the parent, the heterozygous parent in this cross, was in cis linkage for all three loci. So there's an extra bit of information for you. Okay, so now we know what the parents are. So what we're going to do is look at what changes in those parental chromosomes, right? So what differences are there from that three recessive or three positive, three dominant alleles? Okay, that's step one. Step two in your plan for happiness is find the ones with the smallest number of recombinants, which are these. Those are your double recombinants. And so just as the probability of getting one recombination between two loci, or one recombination at all, is X, getting two recombinations on the same chromosome is going to be much rarer than that, right? So you'd expect many fewer recombinants to be the result of two recombination events than just one. Okay, so these are your double recombinants, right, which are listed here. Now what you do is you go, which allele moved? Or which locus had an allele that moved? Okay, and we look at them. We've got lazy, dominant, dominant, recessive, recessive. That's the same. We've got dominant, dominant, recessive, recessive. That's also the same. Well, you know what the answer is because there's only three of them. Now we have dominant, recessive, recessive, dominant. So these recombinant chromosomes have had this gene move. And the only way, what did I have on the next one? No, nothing useful. Well, not in this point anyway. The only way in which you can have one locus move and it be the lowest frequency is if that locus or that gene is in the middle. So if you see here, we've got, again, another individual with cis linkage, or these genes are in cis linkage. And now we look at the double crossover. We go, this dominant and this recessive flip. And so we end up with a recombinant chromosome that's dominant, recessive, dominant, recessive, dominant, recessive. So the only thing that has moved is the one in the middle. So that's what tells you your gene order. So this is wrong, right? This gene order that you're given is incorrect. You have to figure it out yourself. Okay? And then the third route to success and happiness in 3-point crosses is to work out the recombination frequency between those loci. And so the same thing applies with the 2-point crosses. You add the recombinants together, but now, and this is really, really important, or you'll get it wrong, you add the single recombinant. So if we want to find out the distance, and this is now the correct gene order because we've figured it out. If we want to calculate the distance between LZ and SU, we have to add these recombinants together. Remember, they're a mirror image, so we need to include both sets for that single recombination, and the double recombinants, because those are also the result of a recombination event between the LZ-SU loci. You add those together, that's 79 in total, and then you express the recombination frequency as 79 divided by 74, or 740, sorry. And that comes out to, I think that's 10.7%. And you do the same for the other two loci, in this case SU and GL. You add the single recombinants together and the double recombinants, and then divide that number by 740. So for those two genes, it's 109 in total, divided by 740, and that's 14.7. So this is a perfectly good map. Let's see what I've got elsewhere. Okay, so this is just a nice picture. This is another test, right? So this is just one that I made up on the spot. And again, you do the same things. First, what are the parents? These ones up here are the parents. This also tells you that these genes are in cis linkage in the parent. Step 2, what are the rarest recombinants? That's these ones down here, okay? And so that tells you that Q, because if we look at this particular chromosome, dominant R, recessive Q, dominant D, and compare that with this parental chromosome, the only one that's changed is Q. So that has to be the one in the middle. And vice versa, again, because they're mirror images for the other double recombinant. So that tells you that your professor is not playing silly beggars with you, and this is actually the gene order, right? So no trick here, but you have to confirm that yourself. You cannot assume it, ever. Okay, so now we know the gene order is RQD. We know that these are the parental chromosomes. Okay, so what we do now is we add all this lot together. I can't remember what that number is. It's in the chat. Here's my chat. Oh, that's for something else. Whatever that number is. If you want to find out the distance between R and Q, you're going to add these two recombinants together, right? Because these recombinants only result as a result of recombination between these two genes, and the double recombinants. So that's what, 68 over the total. You do the same for the other two. So between Q and D, these are the single recombinants between those two loci. And so you add those together, which is what, 159 plus 8, 167? Yeah, I think that's about right. And again, calculate that as a percent over the total. And so what that gets us is these two possible maps. Forget the A's for a second. But we have a map distance of 18 centimorgans. That looks like centimeters. It's not supposed to. It's supposed to be a capital M. 18 centimorgans between R and Q, and 7.3 centimorgans between Q and D. Again, in and of itself, we only know the relative order and distance of these three genes. So these could be either way around. It could be RQD or it could be DQR on the chromosome. We do not know. We'd need to do another cross or 3-point cross with a fourth locus or gene, let's say gene A, to be able to figure out if it's ARQD or ADQR, right? And so that would be again, and this is essentially how these linkage maps of chromosomes were built. And that's pretty much it. So remember, follow your 3-step plan for happiness. No, I'm not going to tell you to call a certain number and enter your credit card info, but follow those and you can't get it wrong, right? That's a guarantee. Ironclad, right? 12 months warranty. And if you, I would again strongly recommend that you practice these, particularly the 3-point crosses, right? So that you get into the habit of just thinking, okay, I need to do, where's the parentals? Oh, it's this one. Okay, well, what's the double recombinant? Oh, that? Oh, that's the gene order. Okay, well, what's the distances? And you just work it out, right? Because you don't want to see a question like this in the exam and it's just like, poof, how do I do this? Okay. All right, that's pretty much it for me. So again, if you have any questions, please do make a note of them. Come to the WebEx on, or hopefully Blackboard Collaborate Ultra on Tuesday, or shoot me an email if you don't understand, okay? And then I'll be able to answer those questions either in person or by email. All right. Thanks very much, everybody. And again, my apologies for not recording this this morning.